This brief lecture on gender determination using information about genes and hormones is for my medical physiology class during the spring of 2019. Without extensive education in physiology, it might be just common to assume that the two genders, male and female, are just easily determined by looking at an ultrasound when a baby is developing. However, this is not the case. In this class, this semester, we've learned about lots of different hormones as well as different anatomical structures that are both used to determine if someone is either male or female. And this lecture will be a more in-depth view of the difference in development between males and females from both the genes and the hormone perspective. So this is just slides, a brief overview and review that will talk about some information that we've learned in our prior Unit 3 on the endocrine system as well. So as we are all well aware, sex hormones play a role in behavior as well as in brain structure. There are different versions of hormones that are present in both male and females that we've discussed in previous units, such as Unit 3 in our chapter and discussion on the endocrine system that play a unique role in sexual development. These hormones play a role in the development of reproductive organs as well as some brain structure and function. So what's important to remember from last unit is this. So this is the pituitary gland, and it has the anterior parts and the posterior component. So we have different hormones that are secreted from the anterior and the posterior. So what's important for this lecture is the anterior pituitary has the ability to secrete prolactin, which is used to develop breast milk, dopamine for the excretion of breast milk, as well as luteinizing hormone and follicle stimulating hormone that are released to the testes and gonads that are used for sexual development. In the posterior pituitary gland, the only reproductive hormone that we kind of discussed was oxytocin or vasopressin, whichever you'd like to call it, which is used to begin or to stimulate labor, rather. So those are both important to keep in mind as we move forward to kind of get ourselves to start thinking about different hormones in this body system. So sex determination overview. This will be brief, and basically each nucleated cell in our body has 46 chromosomes, 22 matched pairs of autosomes, and 1 pair of sex chromosomes. So autosomes are our body cells, and they direct the development of physical features and blood types. Sex chromosomes, on the other hand, determine the genetic sex of the individual, which is, like I said, either male or female. So that'll be kind of discussed in our next slide. So this is a karyotype, and karyotypes are just a display of what genes and chromosomes actually look like laid out. So males are XY, females are XX, and as you can see, and you probably are already aware, females inherit one X from the mom and one X from the father, and males inherit X from the mom and Y from the father. So when we're talking about the genetic determination of sex, usually what we're talking about is XX or XY, which are just chromosomal developments that occur during fetal development. So even though this is a relatively complex topic to discuss, if we look at it from this way and kind of determine, okay, well, we have the X and the Y, they come together, XY male, XX female. We could take a look at all the different chromosomes and the different regions on each, which if you take a genetics class later on, you'll be able to kind of learn more about different kinds of genetic abnormalities that can present themselves either in childhood or later in adulthood. So we're not really going to talk about that too much. However, it's important to know just the basic genetic structure for this lecture. Sexual differentiation occurs early in development. So in addition to the actual XX or XY developing, we have different hormones and something that we call gonadal bipotential that play a role in determining if a fetus will become male or female. So the gonadal bipotential occurs around the 7th week of development, which is before the differentiation of sex occurs. So you have an embryonic potential that cannot morphologically be identified as either male or female. So the bipotential gonad has an outer cortex and an inner medulla, which is similar to a lot of other organs that we've seen in the body, such as in the kidneys, we have the adrenal cortex located there. And given the appropriate signaling and development, you can have these bipotential gonads develop either into testicular tissue or differentiate into ovarian tissue. And this basically occurs with interactions with the Wolffian and the Müllerian ducts. And if you look over here in this column, we have some genes and hormones that play an important role in male development, which is different from female development. So the SRY is located on the Y chromosome, so females don't have that, and that helps determine male genes. I'm sorry, the SRY gene is what helps determine male characteristics on that Y chromosome. So it's located on a specific region on the Y chromosome. SOX9, WT1, both play a role and are influenced by SRY, and those are only in males. Anti-Müllerian hormone, AMH, and DHT are both male hormones that help facilitate the development of testosterone and the different male reproductive organs. And this is really important because, as you'd expect, female reproductive system does not have this. And this is important because once you have the elimination of anti-Müllerian hormone and DHT, you'll start to differentiate into ovarian tissue. So that's kind of one of the differentiation points between testicular tissue as well as ovarian. So in females, recall that it's the ovarian tissue that differentiates from the gonadal bipotential, as discussed over here. So we have no SRY gene or testicular anti-Müllerian hormone. So what you have is similar, as we've talked about in our anatomy class, anatomy follows function, form follows function. We have Müllerian ducts and Wolffian ducts both present in males. And like I had mentioned before, once we develop from male to female or female to male, we no longer have AMH and DHT in females, which makes sense. So that doesn't mean that the ducts where they're secreted from go away. They're just going to develop into something different, something that we can use anatomically, something that makes sense functionally for females. So we have the Müllerian ducts that develop into the vagina, the uterus, and the fallopian tubes, which are used during, of course, all phases of the female life. However, more importantly, starting at the menarche period, puberty, which is around between the ages of 9 and 13 for girls, and 8 to 14, and then 9 to 13 for boys. And you have the Wolffian ducts actually degenerate due to no testosterone. And similarly, without DHT, which is a testosterone derivative, you actually have the genitalia differentiate from female to male in anatomical appearance. So that final step of the DHT being eliminated actually leads to the appearance of what we would recognize as a vagina. So this is all super important because without these structure and function differences that occur both in embryonic development and while the embryo is developing into a fetus and then into what we recognize more as a baby, before it's born, of course, you have a lot of changes that can occur. And there's even some rare cases where you have individuals born that the sexual parts, like the penis and the vagina, aren't fully developed until later on. So you kind of have to sometimes make a choice as a parent or in a role where you can give the child hormones to kind of help the differentiation process along. So again, not to get into any ethical or political debates, but this is just a practical application and something common that we might see about during a time in America where we do have a lot of people who are either transitioning or it's a topic of discussion. So there's definitely a lot that we talk about today that comes down to this physiological process that occurs very, very early in development. So thanks again for listening.